So we discussed about the simple proteins and also about the conjugated proteins. Simple proteins that are mainly consisting of the only made up of with the only amino acids, one amino acid that are called as the simple amino acid. And whereas the conjugated protein that are mainly containing the prosthetic groups and which are called as the glycoproteins, examples of the N-acetyl acid, arabinose, glucose, mannose, glucosamine, N-acetyl glucosamine. Whereas chromoproteins containing the colored prosthetic groups which are mainly of functioning in hemoglobin, myoglobin, chlorophyll, and also of the rhodopsins. Okay, whereas here the chromoproteins are of mainly hemoglobin, myoglobin, chlorophyll, and rhodopsin. Whereas the phosphoproteins that are of the agophosphatide, and that is mainly phosphoric acid linked to serine and threonine residues, which are the hydroxyl group containing amino acids. And nucleoproteins or the simple basic proteins, which are of the protamines and histone, which are the protamines of histone. These are combination with the salt and that contain nucleic acid as the prosthetic group. And these are the main important constituents of the chromatin structure that are called as the nucleoprotein. Mucoproteins means that are containing the mucopolysaccharides, which are mainly of the hyaluronic acid and chondroitin sulfate, and that means these are mainly yielding about the amino sugars on their hydrolysis. Okay, and here lipoprotein that containing the lipids such as fats, phospholipids, and cholesterol. And metalloprotein that containing the metal binding proteins. Here, the transferrin is mainly combining with the metals, that is iron, copper, and zinc. And also celluloplasmin, which contains the copper element. And derived proteins means these are mainly formed due to the hydrolysis action on simple or conjugated protein. And what are the derivatives that are formed means that are called as the primary derived proteins and secondary derived proteins. And the primary derived proteins that are mainly formed by slight changes in the protein molecules and its properties. And here there is no hydrolytic cleavage of the peptide bonds here. There is no cleavage of the peptide bond. Whereas the secondary derived proteins that are mainly formed by the cleavage of the peptide bond. Here, the main form of derivatives of the primary derived proteins are the proteins, metaproteins, and coagulated proteins. Okay, and these are mainly formed by the action of the heat or alcohol. Whereas the metaproteins that are mainly formed by the action of acids and alcohol. Whereas the proteins that are mainly formed by the action of water, acid, enzymes. That means here the primary derived proteins that are not formed by the hydrolytic cleavage of the peptide bond, but are mainly formed by the action of water, acid, enzyme, or that means that is depending upon the solubility nature also, and also depending upon the action of heat or alcohol, these are formed. And secondary derived proteins that are mainly due to the cleavage of the peptide, hydrolytic cleavage of the peptide bond. These are mainly formed due to the hydrolytic cleavage, and which are of proteases, peptones, and peptides. And these are the main derivatives that are formed due to the action of the hydrolysis on peptide bonds and also on due to the action of the without on the action of the this peptide bond. And coming to the structural conformations of the protein, and these are mainly existing as the primary, secondary, tertiary, and quaternary conformation. And here, the primary conformation that is mainly occurring in the linear form and which are formed by peptide bond, whereas secondary conformation that occurs in the form of helix and shape, and these are mainly formed by the peptide and hydrogen bond. Whereas the tertiary structural conformation that is mainly formed with the all type of the interactions like electrostatic, ionic interactions, and also the sulfide bond formation. Whereas the quaternary conformation here, the proteins which are formed as the monomeric units that are combined together form the polymeric units which are formed by the result of quaternary structure. And before that here, first of all, we have to discuss about the peptide bond. And what is peptide bond means here? The carboxyl group of one amino acid and that is combining with the amino group of the other amino acid that resulting in the formation of the peptide bond which is called as the CO, C double bond O NH bond. And in this, depending upon the number of amino acids combining, and these are defined as the number of peptides. That means here, for example, two amino acids are combining means, and that are formed in the formation of the dipeptide bond. dipeptide bond. Whereas the three amino acids are present, and that means that is involved in the formation of the tripeptide. But here, and how many peptide bonds are involved means in dipeptide, here only one peptide bond is there, and in tripeptide here two peptide bonds are present. That means here it is describing here that is describing only number of amino acids. Okay, and here the multiple amino acids and that are resulting in the formation of the polypeptide. Here the free amino group and the free carboxyl group and that are resulting in the formation of the peptide bond. This is the peptide bond. Here hydroxyl group and this amino group and that is involving in the bond with the releasing of the water molecule. Okay, and here whatever the bond that is formed and that is the amino acid that is combining with the peptide bond and that is forming as the backbone of the protein. Okay, and here the peptide bond is written as a single bond. That means it can be written as a CO or CN bond. And because of the presence of the resonance between that here, and that can be written as a C double bond ONH also. And this is mostly present in trans conformation. This is present in trans conformation, and it is also called as the coplanar. And that means there is no pre-rotation around the CN axis. Okay, and that is called as the coplanar conformation. And here the primary structure of protein, and that means it is mainly forming the simple sequence of amino acids, and that is mainly formed by the covalent bonds linking the residues together. And the minimum size of the protein that is containing the 50 residues, and these are forming the peptides. And here, and that have the molecular weight of the 5,000 Dalton. And there is no here maximum size, but the largest protein that is discovered has about the 30,000 residues. And average molecular weight of the residue is about a 110 Dalton. And you have to remember this also. This is the most important question. Average weight. Average molecular weight is 110 Daltons. 110 Daltons. Okay, and next coming to the secondary structure, and this is mainly formed by the folding of the polypeptide backbone, and that is mainly formed by the, it is stabilized by the hydrogen bonds between the NH and C double bond both. And here various secondary structures are present, and that are forming the helical structures and beta sheets. And here these are the structures here, and the helix that is present in the conformation of the helical conformations here. And pleated sheets means that are forming like sheets like structures. Sheets like structure. Okay, and these are the helical structures and sheets. And here we can also observe the tertiary structures here, and that is mainly formed by the different type of the interaction. That's the quaternary structure that are mainly formed by the combining of the units. And here in alpha helix, in this conformation, the helical arrangement of the polypeptide chains occur that are forming the coiled spring. And here the carbonyl and NH groups that are oriented parallel to the axis, and that are forming the alpha helical structure. And these have the dimensions of 3.6 residues per time, and that having the 0.4 nanometers per time. Whereas the structure of beta pleated sheet that is forming, that is different from the structure of alpha helix. And here in beta sheet, the polypeptide chain folds back on itself so that polypeptide strands like side by side. Here, the side by side structure, the side by structure is present, and that is linked with the hydrogen bond, and that forms a sheet like structure. Okay, and here, and that means here these bands are mainly formed between the neighboring polypeptide strand. So these are from the beta sheets. Beta sheets. Okay, that means here the primary structure folds back in the formation of the parallel or anti-parallel arrangement, and that are producing a parallel or anti-parallel beta sheet. And depending upon the arrangement of the amino acids here, that are forming the sheets and helical structure, and that are mainly forming the secondary conformation. Secondary conformation. And here, how the amino acids are folding here means that is mainly depending upon the steric hindrances that are existing between the molecules. Steric hindrances or the charge repulsion, and that are mainly present in between the different type of the amino acid. Depending upon that, here the conformations are present. Okay, coming to the tertiary, and this is a 3D arrangement, three-dimensional arrangement of the proteins. Okay, and here these have the particular pattern of folding, and whereas here the secondary structure that is stabilized by hydrogen bond, and also weak pulses that are contributing the tertiary structure, and that are of the hydrophobic interactions and also of the electrostatic interactions. Okay, and that are mainly found here. And here these contain both the hydrophilic and hydrophobic residues, and that means here the hydrophobic parts are that are mainly present at the center, whereas the hydrophilic parts are present on the exposed to water. And that means here, and these are containing, these are forming the amphipathic proteins, amphipathic structures. Okay, other forces that are mainly forming are here ionic bonds, hydrogen bonds, and van der Waals forces. And these bonds are weaker than the covalent bonds. And also here, one is the covalent bond that is involved here is disulfide bond, and that is mainly formed between the cysteine residues. Okay, and this is mainly present in non-cytoplasmic protein, which where the enzymes are present in the cytoplasm proteins to remove the disulfide bond. And these are the 3D structures that can be visualized by observation here and here, and that are mainly formed by the hydrogen bond, disulfide bridges, ionic bond, water wall interactions, and hydrophobic interactions also. And next coming to the structural domains of the protein. And here the structural domains, here based on the domain classification, here these are considered as a quaternary structure of protein. Here what is that means the main level of organization, and that is giving to the domains. And here the domain is a distinct region, it is a sequence of amino acids of protein, and that is independently folded part, and that forms a stable structure that all called as a domain. Here, the quaternary structure, and it is mainly composed of the more than one polypeptide chain. And depending upon the number arrangement of the individual polypeptide chain, these are formed as a subunits, and that subunits, and that are linked up with each other, and that forming a complete protein. And these are forming also with the identical or non-identical subunits also. And depending upon that here, and these are forming as the protein. And here, for example, when we take the antibody, antibody contains the two larger subunits and two smaller subunits, which are called as the heavy chains and light chains, which are connected by disulfide and non-covalent forces. And that are called as the quaternary structure of protein. And here we can see observe the, and these are the alpha and beta chains of these are the alpha and beta chains here, which are intertwined with each other, which containing the heme acid cofactor. And here, and that means here the hydrophobic forces are present at the center, whereas the hydrophilic forces that are exposed to outside. And here the protein folding, and these are mainly forming these conformations. Okay, and these the protein foldings are, and that can be undergo the process of the denaturation, and that can produce the primary, secondary, and tertiary conformation. And that means these are mainly disrupting the bonds in between the amino acids that are mainly released in the primary, secondary structure. And sometimes, and these are really reunite together, and that process is called as the renaturation. Here, denaturation means breaking, renaturation means uniting. Okay, and that are forming different type of the conformation centers.